Have you ever wondered how DNA replicates? Or how the process of protein synthesis works? Well, hopefully today I will give you a better understanding of both. Let's start with DNA structure and the basics. DNA stands for deoxyribonucleic acid and is found in almost all living things. DNA is the carrier of our genetic information. Inside of DNA are two chains that twist and coil around each other. They form a double helix. These helicases are also known as the twisted ladders of DNA. DNA is stored in the nucleus. The cell cycle. First, in the G1 phase, the cell gets larger. Next, the DNA gets unzipped by the enzyme helicase. Polymerase then copies this. Eventually, you will end up with two identical gene pairs. In prophase, the cells are released from the nucleus. In metaphase, they align in the middle. Next, anaphase pulls the cells on either side. Then the telophase places chromosomes in a new nucleus. Finally, calisthenics comes and divides the cells into two new cells. Now let's talk about the replication of DNA. The replication of DNA is the biological process of producing two identical replicas of DNA, from one original DNA molecule. There are three steps in the replication of DNA: initiation, elongation, and termination. During replication, the ladders split in two and this process is called the replication fork. From there it then begins to copy itself and pair with another set of bases. An A base can only bind with a T base and a C base can only bind with a G base. The base sequences must complement each other. Now onto protein synthesis. Protein synthesis is the process where individual cells build their specific proteins. There are two steps in protein synthesis: transcription and translation. Transcription is where the information is encoded into DNA and then encodes that information into mRNA, messenger RNA. During translation,